Reinnervate was originally a spin-out from Durham University in the UK. It was founded in 2002 by Professor Szyborski, really in an effort to solve some of the intractable problems he had in cell biology, namely how to grow cells in vitro so that they provided a better proxy for in vivo cell growth.
The study of cells in conventional 2D culture systems is not always truly representative of cells within a living tissue. To address this, we have developed a technology known as alvetex, which enables cells to grow readily and routinely in three-dimensional cell culture. And they acquire a more natural shape and behave in a way which more closely mimics cells in living tissues. It enables researchers to create more representative model systems, to study disease processes, basic principles of development, and test various biological processes. Ultimately, we hope that the use of alvetex will reduce product development costs, particularly in the pharmaceutical industry, and also have a positive impact on reducing the number of animals used in research.
The power of routine 3D cell culture is that it will drive new research, different research, better research. It'll enable cell biologists to do things that hitherto they've not even thought of. And we know this from talking to cell biologists in commercial and academic environments. They are looking to unlock the power of their cell-based assays. We realised that to have broad appeal, alvetex had to have certain attributes. First of all, it had to be relatively inexpensive. Secondly, it had to be easy to use and transferable. You can use alvetex in any laboratory where you currently do cell culture. We also knew that it had to enable robust and reproducible scientific experiments. And these are all the attributes that we've developed and built into alvetex today.
Alvetex is a highly porous polystyrene scaffold with a well-defined and uniform structure which has been specifically engineered for three-dimensional cell growth. Within the human body, the majority of cells are no further away than 200 microns from a capillary. So we've specifically engineered alvetex to that thickness to enable good transport of nutrients in and removal of waste products, and in that way to ensure healthy and viable cell growth. Alvetex is manufactured in our purpose-built facilities, including our Class 10,000 clean room, to the principles of good manufacturing practice. Alvetex is available in standard laboratory multi-well plate formats, as well as some specific well insert formats.
Cells will enter alvetex and they will form three-dimensional relationships with other cells. Given time, cells will proliferate and fill the scaffold, creating, in effect, a micro slab of tissue within the cell culture vessel. A wide variety of standard cellular and molecular techniques and biochemical assays can be applied to the study of cells grown in alvetex. We have used alvetex technology to demonstrate that when growing hepatocytes in 3D, we can get enhanced cell structure and function. This will lead to the development of more relevant safety and toxicity assays. Alvetex has been exemplified using skin keratinocytes isolated from the epidermis. We've demonstrated that we can generate stratified layers of skin cells, including the formation of the stratum corneum. These are important for the development of cosmetics and pharmaceuticals and for use in skin studies and epidermal barrier assays. We have grown osteoblasts. We find that the cells readily form bone nodules. They produce large amounts of extracellular matrix, including collagen.
Alvetex has been developed with a great deal of painstaking thought and effort. It has been rigorously scientifically exemplified, as anybody that's been to our website will know.
We've published widely on this subject, have generated data which has been subjected to peer review and have presented our findings at a variety of international conferences.
Alvetex is available globally today through a network of regional and global distribution and commercial partners. So now, for the first time, scientists everywhere can unlock the potential of three-dimensional cell culture in their laboratories.